Good evening, class. Welcome to week 10 of Living Systems. Not long to go until the end of semester, and tonight we'll be looking at some plant anatomy. Lecture 14, structure of plants, and then after that we'll cover a little bit of lecture 15 and we'll finish that off next week. So let's have a look at plant cell walls, which you've already been introduced to. So you're familiar with the fact that cell walls define the size and shape of plant cells. They contribute also to metabolic processes and cell-cell communication. Processes that you'll learn, or some of you will learn in more detail next year. Lying in between adjacent plant cells normally is a region called the middle lamella. And this is made of a substance called pectin, which binds the cells together in the same way that you would use mortar to bind bricks together. And we'll look at that on the next slide here. So this is a cross-section of a certain type of cell from a stem, and we'll look at the structure of these cells here. So we've got several cells. This one here has just been raised up above the plane of the other cells so that we can see this outer cell wall, and you'll see that it's called a primary cell wall. And to the inside of that, this thin brown region, this is the plasma membrane, and to the inside we can see the nucleus and that large central vacuole. But what I wanted to point out to you is the middle lamella. So you can see that dark purple region in between the cells there. Okay, so plant cells are surrounded by a cell wall, and most of them are surrounded by what's called a primary cell wall. Some cell types in plants also have another wall layer, which is called a secondary cell wall. And I'll show you an example of that type of cell later on. So the primary cell wall, this is the first cell wall layer formed by a new developing cell. And we saw last week that it's composed of cellulose microfibrils together with pectin molecules, other polysaccharides called hemicelluloses, and there's a few different types of those, and also various proteins. The primary cell wall is quite thin and it's quite flexible, which means that it can extend as a cell enlarges and matures. These primary cell walls vary in thickness. There are thinner areas called primary pit fields, and within these primary pit field areas there are numerous plasmodesmata. And we introduced the concept of plasmodesmata. You might remember that they're those really tiny channels that connect each cell to neighbouring cells. So here we have a cell here showing the primary wall, the thick region of the primary wall, and then as we come around here you can see that it's quite thin. So this is the primary pit field, and you can see some little faint lines there. These are the plasmodesmata that are linking this cell here to the neighbouring cell. And once again, this line here represents the middle lamella joining those cells together. As cells mature, certain cell types will lay down a secondary cell wall, and this is laid down on the inner surface of the primary wall. So let's just draw that. Here we have the primary cell wall and the plasma membrane, and then the secondary wall is laid down in between the two. The secondary cell wall is usually formed in 3 distinct layers, each with cellulose microfibrils laid down in different orientations. And the secondary wall, unlike the primary wall, is strengthened by the addition of a substance called lignin. Lignin belongs to a group of organic compounds called phenols. Some cells have many depressions or primary pits in the secondary wall, just like the primary walls do. All right, now let's revisit our plant tissue systems. And we introduced these last week in the cells and tissues lecture. The primary plant body is composed of cells that are produced by shoot and root apical meristems. So let's recap our primary meristems. So here's the soil surface, and here's my terrific drawing of a plant, and here's the root system. Okay, and of course we have a region of cells here at the tip of the shoot, and this is called the shoot apical meristem, or simply SAM. And then down here, just behind the very tip there, we have a group of meristem cells, and that's referred to as the root apical meristem, or RAM. And when you think about meristem cells, or meristematic cells, as they're often referred to, think about stem cells in animals. They act in the same way. That is, they're undifferentiated, and they will divide to produce new cells, and some of those new cells will differentiate to form certain tissues of the plant body. Now remember, these are primary meristems, and primary meristems tend to add height to the plant and, of course, depth to the plant. And we'll talk about secondary meristems a little bit later. All right, so first of the plant tissue systems is dermal tissue. And the major type of dermal tissue that we see is epidermis. Epidermis cells segregate the internal plant tissues from the external environment. Epidermal cell structure varies considerably depending on whether you find it in the stem, root, or leaves. Although the main function of epidermal cells are in protection and facilitating exchange of materials. But you'll come to see that there are some epidermal cells that have very unique functions. There are two very unique types of dermal tissues, including the endodermis, which we'll come back to, and also the exodermis. So keep those two in mind. Oops. I always forget to get rid of my drawing. There we go. All right, second major plant tissue system is ground tissue. And these tissues vary considerably depending on where they're found. They include parenchyma, collenchyma, and sclerenchyma, and also other types of ground tissue systems. These are just three of the major ones. Ground tissue makes up the bulk of tissues in stems, roots, and leaves. So let's start with some examples of parenchyma cells. Here are parenchyma cells found in potato tubers. And these cells here have been stained with a particular dye that specifically stains for starch. And hence you can see all these black starch granules within the cells. So obviously these parenchyma cells are specialised for storage. Another type of parenchyma cell is the erenchyma cell. And erenchyma cells tend to have these very large air spaces. So you can imagine that tissues that require large air spaces may be in an oxygen limited environment. So, for example, plants that are always waterlogged or tend to inhabit aquatic environments. Another type of parenchyma cell is the transfer cell. And these cells tend to have all these ingrowths of the plasma membrane and cell wall. So they look like little lines, but they're actually ingrowths, wall ingrowths. And these ingrowths function to increase the surface area of the cell, which enables really rapid transfer of molecules from one cell to the next. All right, so parenchyma, we've just seen some examples of parenchyma cells. Parenchyma tissues normally comprise large cells with thin primary cell walls and a well-defined pectin-rich middle lamella. Here are some different types of parenchyma cells. We just looked at storage parenchyma, which specialise in storage of nutrient reserves. We've looked at aerenchyma, spongy cells with large network of air spaces adapted to low oxygen environments. Transfer cells, which have all those ingrowths that increase the cell surface area for rapid transfer. And here's another type, a very important type, chlorenchyma cells. These are the photosynthetic cells of the plant, found in leaves and also in stems. Right, collenchyma, ground tissue. This is supportive tissue, providing strength to plant parts where bending and flexibility are required. Sclerenchyma tissue. This is also supportive tissue that imparts strength, but rather than flexibility, it imparts rigidity. Here are two major types of sclerenchyma cells, fibres, which I'll show you a little bit later on, and sclerids. Now, one thing to note about sclerenchyma cells is that these cells, after they develop, actually die. But even though they die, they still carry out the role that they're intended to carry out. So we say that these are dead cells at functional maturity. Okay, our next major tissue type is the vascular tissue. And I often compare the vascular tissue in plants to the blood circulatory system and lymphatic system in animals. There are a lot of similarities between the two systems. Vascular tissues in plants are involved in the transport of materials, of course, around the plant body. The two major types of vascular tissue include the xylem tissue and the phloem tissue. Let's recap what we know about xylem and phloem. Xylem transports water and dissolved minerals from the roots up in an upwards direction to the leaves and the rest of the plant. The common cell types seen in xylem tissue are vessels and trachees, and we'll come back to those in just a minute. The phloem vascular tissue transports the products of photosynthesis, from where these products are synthesized primarily in the leaf to actively growing areas in the plant or to storage. So the products of photosynthesis, let's think about what these products are. Of course, they are carbohydrates, but we'll start referring to them now by the correct term, which is photosynthates. So let me type that in there. Photosynthetases, products of photosynthesis. All right, so let's now start with the xylem vascular tissue. And we'll look at one type of cell, one of the major types of cells in the xylem, and these are called vessel elements. These are common in flowering plants, They're quite unusual cells in that they're not fully enclosed. They have open ends that allow an unimpeded flow of water from one cell to the next. So let's look at that here. So here's one vessel element and here's a second one. So you can see they're stacked on top of each other. They look more like little tubes than what you would typically expect to see from a cell. And here's the open end of one of the vessel elements. And you'll see the same thing at the other end, if you could see that properly. All right, so that's these are two young vessel elements. Here we have two large mature vessel elements. And of course, the blue arrow is showing the direction of water and dissolved nutrients, the direction of flow. Now, one unusual property of vessel elements is that they always develop that tough secondary cell wall, which is impregnated with lignin. Another property of vessel elements is that they are also dead cells at functional maturity. They actually lose most of their cell contents, and as a result, they have to be supported by other cells, cells that are metabolically active. And those support cells, if you like, are shown here. These are xylem parenchyma cells, also part of the vascular tissue. But they're more support cells rather than transport cells. So again, here's one of our vessel cells or vessel elements, and here's a younger vessel element nearby. So you can probably imagine why it's necessary for these cells to be strengthened by the development of the secondary cell wall. Any idea why these cells have to have that tough secondary cell wall?
Like I said, continuously exposed to fluids and stuff like that.
Yep, so something about those fluids. So imagine these tiny little tubes, I call them tubes, but they're cells, tiny little tubes, all stacked on top of each other and extending right up through a tall tree. And the water's moving up through it. Imagine the type of pressure that they would be exposed to in order to move water and dissolved ions and nutrients all the way up to a tree that's maybe 100 metres tall. We're talking about some enormous pressures here, and hence the requirement for secondary cell wall formation and strengthening with lignin. Okay, here's our second type of xylem transport cell, and these are called tracheids. And tracheids are common not just in flowering plants, but common in all plants that have a vascular system. Tracheids, unlike vessel elements, have intact end walls, and as a result of that, the water that moves from cell to cell moves through tiny pores called bordered pits that are found in the cell wall. So let's have a look at that. Here's one tracheid here. So they're fully enclosed cells, but part of it's cut away here. And here are the bordered pits. There's one little bordered pit there and one there. And here are some more bordered pits showing the movement of water from one tracheid to the next. Moving on now to the second major part of the vascular system, the phloem. The major cells found in the phloem tissue are sieve tube cells. These are the transport cells found in phloem tissue. They are associated with a helper cell, or rather a few different types of helper cells called companion cells. Now, sieve tube cells, unlike those xylem vessel elements, are living cells, but they are quite reduced in their cell contents. So they still require that metabolic support from these helper cells. Sieve tube cells are elongated. They possess thin primary cell walls, so they don't have secondary cell walls. And of course, primary cell walls are composed largely of cellulose. The companion cells control the activity of sieve tube cells. And here is an illustration of a sieve tube cell, and you can see where the word sieve comes from, because each end of the cell looks like a sieve, and this is actually referred to as a sieve plate. So it's an intact or enclosed cell, but we have these large pores found on each end, and they also stack on top of each other, as you can see here, two cells stacked on top of each other. You can also see these groups of pores in the side wall of the cell. They're called sieve areas, and this enables the movement of photosynthase also in a lateral direction. So these sieve tube cells normally are clustered together in bundles. So those photosynthetes are moving not just in an upwards and downwards direction through the phloem, but also laterally to any cells to the side that require photosynthetes. And here we have a helper cell. Here's a companion cell. They're also very closely associated with these cells. And here's another illustration of a companion cell. All right, let's start with a closer look at stems. But before we do, here we have a nice overview of a flowering plant, except that there's no flowers shown here. So let's just look at the structures here. So the entire part of the plant above the ground, of course, is referred to as the shoot system. Below ground is the root system, although, of course, there's always exceptions to any rule. So let's start with the root system. Right at the bottom here, we have a region of cells referred to as the root cap. Just behind that is the root tip, and within the root tip, we find the root apical meristem, the RAM. Okay, behind that, we'll find a region of the root where root hairs develop. We'll come back to those also. Then we have these root branches, which we call lateral roots. Then here we can see the start of the vascular system. So this is a central bundle of vascular tissue that runs up through the centre of each root. And that vascular bundle contains both cells of the xylem and cells of the phloem, all clustered together, runs up here until it gets to the base of the shoot system, and then it starts to branch off, as you can see. So you can only see two branches here. Normally you would have several branches running up through the stem. And then when one of those branches reaches a leaf, it branches off into the leaf, and this major line that you can see here, this is called the midrib. And then we have smaller veins of vascular tissue running through the leaf. So of course these are obvious. When you look at the leaf, you can see all those little veins of vascular tissue running through the leaf. All right, a few more terms. All right, we have some side stems developing, some side shoots. These are called axillary buds. Here we have the stalk or stem of the leaf. This is called the petiole. The blade of the leaf itself is referred to as a lamina. Lamina is the actual blade. Here we have the point of attachment of the leaf to the stem. That's referred to as a node. So we have a node here, got another node there, and then we've got the region of stem in between two nodes. That's referred to as the internode. And then, of course, right up here within the shoot tip, we have the shoot apical meristem, or SAM. Okay, so plant shoots comprise stems and leaves. The stem provides support, contains vascular tissues, and in some species is a storage organ. So let's look at stems in a bit more detail. And before we do, we're going to just consider flowering plants, which of course are of particular interest to agronomists, agriculturalists, viticulturalists, and so on. Flowering plants are divided into two groups. The dicots, which is short for dicotyledons, dicots, and monocots. And there's a set of characteristics that goes with dicots and monocots, which we won't go into today, apart from one characteristic. All right, so here we're looking at a cross-section of a stem from a dicotyledon, from a dicot. And within this section, we can see vascular bundles consisting of vascular tissue. There's one, there's another one, and within each of these vascular bundles, we find both xylem and phloem. And you can see how they form a ring around the inside of the stem. And this is one characteristic that's unique to the dicots. So if we were looking at a monocot, the vascular bundles would just be scattered randomly throughout the actual stem without forming a ring. Okay. All right, so let me clear that because it's already getting messy. All right, so within each vascular bundle, we have xylem, which is found towards the inside of the vascular bundle, and phloem, which is found towards the outside of the vascular bundle. All right, this ring of vascular bundles and all the cells within it, which you can see are referred to as pith cells, collectively is referred to as the stele. In between the stele and the outer epidermis cells, we find cells of the cortex. And we can see here that there is a single layer of epidermis or epidermal cells to the outside of the stem. Oh, there it is there, epidermis, outer layer of the stem.
Sorry, Silvana, could you just recap on the cortex, please?
Sure, yes. So the cortex, so you can see this ring of vascular bundles here, and all these cells in between that ring of vascular bundles and the outer epidermis, these are referred to as cells of the cortex. It's just a general term, basically, for the cells found to the outside of the vascular bundles.
Got it. Thank you.
Does that make sense? No worries. Okay, so we've seen that the vascular tissue often forms a single ring within the stem of a dicot. It separates the stem outer cortex, that is the ground tissue, from the inner pith cells. The primary phloem forms outside the xylem on the same radius. The earliest forming xylem is called protoxylem, and to the outside of this develops larger, thicker walled xylem called metaxylem. Not particularly important for you to remember, but that's illustrated here. So once again, we have a single vascular bundle here. This is the outside of the stem. These are the epidermal cells here. And within the vascular bundle, we have phloem cells here. And then we have xylem down here. And you can see these larger, thicker walled cells of the metaxylem and the smaller thinner walled cells of the protoxylem. But what I wanted to point out here are cells in between each of the bundles, which are called fibres. And remember, we said that fibres are also represents a type of cell that is dead at functional maturity. This particular type of fibre cell are called interfascicular fibres. Okay, what we've been looking at so far in that stem is what's referred to as primary growth. But stems of some plants undergo a different type of growth called secondary growth. So let's consider those plants. So woody dicots, such as trees and shrubs, can undertake secondary or radial growth. So remember that we said that those primary meristems, the SAM and the RAM, enable a plant to get taller, enables the root system to grow more deeply into the ground, but secondary growth enables a stem to become larger. Okay, so we're talking about adding girth to the stem. And this secondary growth takes place as a result of two different types of meristems, which are referred to as cambium. Just to confuse us, instead of calling them meristems, they're referred to as cambium. These cambiums or meristems produce sheets of cells laterally. There are two major types involved in stem secondary growth. One is called the vascular cambium, which will give rise to wood, and one is called the cork cambium, which gives rise to bark. So I'll try to refer to cambium as cambium meristems, just so you remember that we're still talking about different types of meristems. All right, let's start with the image on the left, and we're going to visit one of those cambium meristems, which is found within this trunk. It's found along this line here that you can see extends right around the inside of this trunk. That's one type of cambium, and it's called the vascular cambium. If we enlarge this little boxed region here, then this is what we can see, these different cell types here. So let's start with these three layers of cells in the middle here, and they represent the vascular cambium. Vascular cambium meristem. So remember, meristem cells are undifferentiated cells, just like stem cells. And to illustrate what they do, I just need to draw a couple of these vascular cambium meristem cells, just like that. Okay, so let's imagine this cell here divides into, there we go, it's divided into two cells. This new cell here remains as part of the vascular cambium, that is, it doesn't differentiate, but the second cell here, this will actually differentiate to form secondary phloem. Yes, here. Now, if we look at this lower cell of the vascular cambium meristem, that will also divide to produce two new cells. Once again, one cell remains undifferentiated, but the second cell becomes differentiated to form secondary xylem. Okay. So here are some xylem vessels, secondary xylem, and some secondary phloem cells. So the phloem cells, secondary phloem is developing in this region to the outside of the vascular cambium, and the secondary xylem is developing on this side towards the centre of the actual stem. So now that you understand that process, we can actually talk about what causes these growth rings that you see inside the trunk of the tree. And of course, they're often used to very roughly estimate the age of the tree. So we call them growth rings. Here they're referred to as annual rings. That's the same thing. And all those growth rings basically arise because of the way the secondary xylem is actually formed. So to illustrate that, I need to do some more drawing. All right, so let's say this is the outside of the stem. And here we have, I'll draw it just further in here so I've got room. Let's say it's the start of the growing season and some secondary xylem cells are formed. And because there's rapid growth at the beginning of the growing season, the cells that are produced are quite large and they have thin cell walls. Okay, rapid growth, large cells. I better start drawing them as circles because I can do that faster. Then towards the end of the growing season, temperatures start dropping, the days become shorter, maybe there's water deficits, nutrient deficits. So now the cells get smaller and smaller with thicker walls towards the end of the growing season. All right, then growth might stop or slow right down. Then we hit the new growth season. Again, fast growth, large secondary xylem cells. And that continues. There's a new layer of cells that's put down, and then we hit the end of the growing season and they become smaller and smaller. Okay, so what you can see here is a very obvious contrast between the growth of one season and the cells that grow in the following season. And that contrast that we can see here is what forms these growth rings, or what you see as growth rings within the actual trunk. All right, does everyone follow that?
Yep.
Yep.
Even with my very poor drawing.
No, they're fine.
Still okay to follow, I hope.
Yeah.
All right, so before I move on, so that means that this growth ring here, this was due to the growth of xylem cells, secondary xylem cells in the last season. This one was the season before, and so on. So the oldest ones are closest to the actual middle of the trunk. Okay. All right, so we've just looked at the vascular cambium meristem. This is a multi-layered cylinder of meristem, and what we saw is that it contained 3 cell layers. It lies between the outer cortex and the inner pith cells, and it's responsible for producing secondary vascular tissues, such as secondary xylem and secondary phloem. Now let's look at the second of the secondary cambiums, and it's called the cork cambium. And these meristematic cells generate the outer covering of the stem or the trunk, and that outer covering is called the periderm. All right, let's look at that here. So here we have the cork cambium in this region here. So about 3 cell layers of cork cambium meristem, and they divide in much the same way that the vascular cambium meristem divides to produce one cell type on one side and a different cell type on the other side. So cork cambium cells will produce cork cells that you can see here towards the outside, and they divide to produce these thin-walled parenchyma cells towards the inside. Now, keep in mind that these cork cells that are produced towards the outside, while they are living at first, when they're first produced, they very quickly die at functional maturity. And now we can actually look at all those layers, those different cell layers within a tree trunk. So in the very centre here, we have what's called heartwood, and the next layer to the outside of that is called sapwood. Both of them are made up of secondary xylem cells. The heartwood, secondary xylem of the heartwood, is no longer functional. It's been impregnated with different resins and oils and so on. So this xylem, this secondary xylem, no longer conducts water and dissolved nutrients. Sapwood, on the other hand, is still functional xylem tissue. The next layer to the outside of the sapwood, and here it is, this is the vascular cambium meristem. To the outside of that, next layer to the outside is the secondary phloem, which is often referred to as the inner bark. To the outside of the secondary phloem, we have this green layer, which is the second cambium, the cork cambium meristem, and then to the outside we have cork or outer bark. Some special functions of stems. Not all plant stems are aerial. There are also underground stems that are formed by certain plant species, and they're often referred to as rhizomes. From these underground stems or rhizomes, aerial branches or leaves may arise, and they might produce new plants. Or they might serve as food storage, as in the potato tuber, which some people find quite odd, because we look at potato tubers and we think of them as parts of the root, but they're actually underground stems. Some more special functions of stems. Desert plants. In arid and semi-arid regions, leaves tend to be absent or highly reduced. And of course, this is to conserve moisture within the plant. In this case, photosynthesis, the process of photosynthesis is primarily carried out by the stem rather than by leaves. Leaves of she oaks, Allocasuarina species, are reduced, highly reduced to very small scale-like structures found at the nodes of stems. Once again, this is designed to minimise water loss from the plant. Stems of marsh and aquatic plants, such environments are usually waterlogged. So of course you can imagine those parenchyma cells with those large air spaces to facilitate the presence of oxygen would be very prominent in these plants. In saline habitats, leaves are also reduced and fleshy stems take over the function of photosynthesize and also function in the storage of water. And here's a nice photo showing stems of the salt marsh plant, Sarcocornea. And you can't see the leaves there because they are so highly reduced. So the stems, in this case, are carrying out those functions of the leaves. Now moving on to roots. And of course, roots are the underground organs, well, in most plants. They are the underground organs of vascular plants and grow from root apical meristems at their tips. Roots have several functions, including nutrient and water uptake from the soil, anchorage and support. Some plant hormones are synthesised in roots. Some of you will learn a lot more about that in plant physiology next year. And of course, some roots function to store nutrients. Root systems may be branched, which is a common trait of the dicots, or they might consist entirely of what are called adventitious roots. This is a trait found among monocot species. So when we talk about monocots, think about grasses. Most of the grasses are monocots. Let's look at the types of root systems. So here we have a taproot system, and also here with primary roots and lots of those root branches, the lateral roots that we referred to earlier. So this is what you commonly find in dicots. In monocots, you tend to find this structure in the root system. These are called adventitious roots because rather than arising from a primary or taproot, they all arise from the very base of the stem. Hence the term adventitious roots, and they make up the typical fibrous root system that you'll see in monocots. So let's look at the structure of a root. A root consists of an outer epidermis. This encloses a well-developed cortex, that is ground tissue, and then we have that inner central vascular bundle called a vascular cylinder. All right, let's start right down the bottom here, and we can see this group of cells here. This is the root cap. Behind that, we find a region called the apical meristem. So, of course, this is the root apical meristem, or RAM. These are the cells that are dividing constantly to give rise to new cells of the root. Then behind that, we have this region here, which is referred to as the zone of cell elongation. These are the cells that have just been formed and they're still growing and elongating. So this is how the roots actually get longer as a result of these newly formed cells. Then behind that, we have this third region here, because I'm not including the root cap as a region. This third region up here, which is called the zone of cell differentiation. So this is where the cells actually take on their final form and function. To the outside, we can see a single layer of epidermis cells, and some of these cells will elongate, as you can see here, to form root hairs. And remember, root hairs are quite different to root branches or lateral roots. To the centre, we can see the vascular system is forming, that central vascular bundle. Okay, in this region, it hasn't quite finished development, and there's no vascular bundle formed here because these cells are still differentiating. All right, if we take a section through this part of the root and then flip that over so that we can see the inside, here's that central vascular cylinder. The dark region here, which looks a bit like a star, this is the xylem tissue, and in this case, it's referred to as exarch xylem. Then here, so at 4 points here, we find cells of the phloem, and collectively they're referred to as the vascular cylinder.
Silvana?
Yes.
If the purpose of phloem is to transport photosynthase,
Yes, correct.
why in the roots?
Sorry?
What's their function in the roots?
Good question. So the question is, if the purpose of the phloem is to transport photosynthase, then why is phloem found in the roots? Is that correct?
Yes.
Okay, good question. Would anyone like to try to answer that?
I guess.
Why would photosynthase be transported down into the roots?
I guess it's a high growth area, so it would need energy.
Absolutely correct. So think about roots. The root system, of course, it's below the ground, so it can't photosynthesize. There are no cells in the root system that can photosynthesize, but those cells still need a form of energy for respiration that occurs within the mitochondria. And the source of energy is carbohydrates, and those carbohydrates arise as a result of photosynthesis. So the photosynthase must be transported always, not just to every other part of the plant, but also to the root cells.
Got it.
Good question.
I guess I just associate photosynthesis with green.
Absolutely, and you're right.
That makes
You're
sense.
right. That makes perfect sense. It's only green parts of the plant that photosynthesize. Absolutely right. And if you look at roots, there are no green root cells. They're always white or grey. Yes, they don't have root cells, do not have chloroplasts. Because, of course, they're not exposed to the light, and you must have energy from sunlight to drive the process of photosynthesis. So good question. Photosynthase must be transported not just upwards to flowers and other structures, but also down to the root system.
Silvana, can I just throw in a bit from Grazier's perspective?
Absolutely, please do.
Okay, so without the storage of the photosynthesis down into the roots, every time I nature the grass, graze it, the plant would die if it didn't have the energy stores to push back up again.
Absolutely. So when you graze really, really close, and you probably graze to the point where there's no green left whatsoever. Am I correct in saying that happens?
You never do that, Silvana.
You never do. All right, never do that. But what if you do? What if you graze? What if you graze? What if you overgraze a paddock? What do you think allows, in a lot of cases, not always, but in a lot of cases, what enables the grass to keep growing?
Regardless, it's actually the energy stores in the roots.
Yeah, exactly right. Every cell in the plant, whether it's cells of the roots or cells in the leaves, cells in flowering tissues, every cell will store a certain amount of energy in the form of starch. Yep. Every cell will store a certain amount of energy in the form of little tiny lipid droplets, which you can't see unless you use an electron microscope. But there is always some sort of energy storage that's going on in every cell type, regardless of whether it's a cell type that's specialised for storage or not. There are always these, starch droplets, lipid droplets, and they're all forms of energy that can be used at any time. But yeah, very good points there. And I'll just point out now you've just made me go backwards in the lecture.
Sorry, everyone.
No, it's really good because it's just made me think of something. So here we have the phloem, which of course is transporting photosynthase, and you find that towards the outside of a stem or a trunk. So everyone's probably familiar with the term ring barking. Has everyone heard of that, when you ring bark a tree?
Yep.
And when you do ring bark, if you cut through that phloem, which is quite easy to do because it's not far beneath the actual outer bark of the tree, what happens to the movement of photosynthase? So photosynthase is still being produced in the actual leaves, in the canopy, whether we're talking about a grapevine or we're talking about a tree. Photosynthase is still being produced and they're still being supplied to the canopy of the plant. But what happens is that they cannot move down to the root system because of that ring barking, because you've destroyed the phloem here. So what happens is that gradually that tree will start to die because of the lack of energy molecules that are being transported down into the root system. The plant won't wilt because water transport up the plant is not affected. It's still being transported up through the sapwood. So you don't get the wilting effect, but you do get the plant eventually dying because the photosynthase can't be transported downwards to the roots. Sorry, off on a tangent now.
Oh, it's interesting. helps bring it all together.
It does, it does. Absolutely. All right, so there's roots. I think we've covered all this. Yep. All right, so let's just recap and add a bit to that. So the root cap. So let me go back. So remember the root cap, this group of cells here. The root cap comprises large parenchyma cells that secrete a substance, a gel-like substance called mucigel. This is a polysaccharide and that contains a mucilaginous matrix and sloughed off living cells. So in other words, the root cap cells secrete that jelly-like substance, the mucigel. It's thought to actually protect the that very tip of the root as it moves, as it grows and moves through the soil particles. Those cells basically are sloughed off constantly, and they're replaced by new cells that come from the root apical meristem, that develop from the root apical meristem. So what does this remind you of in animals? Not something that secretes a gel, but a cell type that's constantly being lost and replaced by new cells. Any idea? I really like to compare processes in plants to animals because there are so many similarities. It's quite amazing. Skin cells, exactly. Skin cells are constantly being lost. They're constantly being shed. It really reminds me that the process is so similar. And of course, skin cells are always being replaced by skin stem cells. So the same sort of process. All right, the root surface area is supplemented by numerous fine root hairs. Remember, these are extensions of epidermal cells located just behind the growing tip. The soil region around the root hair zone, this is called the rhizosphere. This is where interactions occur between the plant and its soil and the soil environment and with the microorganisms in the soil. A very, very interesting topic, which unfortunately we don't have time to go into. All right, the root vascular system, the root vascular tissues of dicots comprise what's called exarch xylem, and we saw that illustrated on one of the previous slides. Now, let's revisit that central vascular cylinder. The vascular tissue within that vascular cylinder is surrounded by a layer of cells called the pericycle. This is a thin layer of cells from which lateral roots arise. So I'd like you to locate the pericycle here. So here it is, pericycle, and that's pointing to this green layer of cells here. And it's these cells that will start to, cell division will be triggered in these cells, and they will start to develop towards the outside of the root. And when this is growing, this lateral root, it basically crushes all the cells in its path until it emerges from the side of the root. Okay, and then it'll grow out in this direction. So these are the root branches of course, as distinct from the root hairs, which are just extensions or elongated epidermal cells. Outside the pericycle cells that we just looked at lies another important layer of cells. These cells are referred to as the endodermis. And you might recall earlier in the lecture, we said there's some very unique types of dermal tissue. This is one of them, because this is dermal tissue that's not found on the outside of the plant. It's found deep within the root tissues. So let's look at the endodermis. So once again, we have our vascular tissue here in red. In this star shape, we have the xylem, and the blue patches represent the phloem tissues. To the outside of that, so this yellow layer of cells, this is the pericycle. And then to the outside of that, and we can see little bits of red in this cell layer, this is the endodermis. Okay, the endodermis cells, and here we have 3 of those cells enlarged. They're quite unique in that they're surrounded, well, on one wall, the radial wall, by this strip called the Casparian strip. The Casparian strip is impregnated with suberin, a very waxy substance called suberin. And suberin is a compound that's impermeable to water. So let's look at how that works. We're going to consider a portion of the root here and look at how water moves into the roots, and then the role of the endodermal cells. Okay, several pathways that water can follow. In fact, when you really think about it, water can enter the root through every possible pathway that exists, but they've just been given names. Let's start with the apoplastic pathway. Water can move into the root, so it's not actually entering the root hair as such, but it's travelling through the cell wall, or in between the cell wall and the plasma membrane. And you can follow this green line here, and you can see that water is never entering the actual cells, the cortex cells of the root. Okay, follow the green line until we reach cells of the endodermis. Okay, we'll leave it there for now. So that's the apoplastic pathway. Here's our next pathway, the symplastic pathway and the transmembrane pathway, which are 2 closely related pathways. All right, let's look at the symplast. So we'll follow this black line here, and you can see that now water is actually travelling within each cell, and it moves from one cell to the other through plasmodesmata. Remember those little channels that connect the cells? So here's the symplastic pathway until it reaches the endodermis. Transmembrane pathway is almost identical, so we'll follow this blue line. Water and dissolved nutrients are moving through each cell, so from cell to cell, but rather than moving through plasmodesmata, water is simply crossing directly through the cell wall and plasma membrane into the next cell. And again, it moves into the root until it hits the endodermis cell. Now, regardless of which pathway the water is actually travelling, once the water reaches the endodermal cells, water can no longer pass in between cells. That means it can no longer pass, it can no longer travel via the apoplastic pathway. At the endodermis, all the water is forced through the symplastic pathway. And this enables water to selectively control the movement of water and ions, and most importantly, it prevents loss of sucrose So sucrose is transported in the phloem. It prevents the loss of sucrose into the roots and out into the soil solution. Okay, so one way of actually protecting those valuable photosynthates in the phloem. Okay, there's a lot more to this story, but we will leave it there for now. Okay. Special functions of roots. Roots of aquatic plants can be very unusual. For example, the mangrove species pictured here often possess upright aerial roots called pneumatophores that function in gas exchange for aerobic respiration. And we can see those here. Very unusual plants, mangroves. Roots and salinity. So our very own Australian river red gums, which are mildly salt tolerant, are tolerant of salty soils because they have this unusual dermal tissue called exodermis. But it's not found on the outside of the plant. It's actually found just below the epidermis. Specialised cells that also have that suberized cell layer, just like the Casparian strip. And this Casparian strip acts as a barrier to the uptake of toxic chloride ions. So this is just one mechanism whereby some plant species are tolerant of saline soils because they actually exclude or prevent the entry of salts into the roots. Special functions of roots, storage roots. So some roots are supremely adapted for storage, and they often have additional layers of vascular cambia together with associated storage cells. A couple of examples, sweet potato and beetroot. And now we move on to leaves, the last of our major organs. And of course, leaves are the primary photosynthetic organs, well, in most plants anyway, with limited lifespans. A leaf usually comprises upper epidermis and lower epidermis. And then within the leaf, we find mesophyll tissue, mesophyll cells. And these are the ones that specialise in photosynthesis. That is, they contain chloroplasts, organelles called chloroplasts. The epidermis is usually covered by a layer called the cuticle. This is not a layer of cells. It's just a substance that's secreted by epidermal cells. So that's one special function of some epidermal cells, to secrete this substance called the cuticle. And the cuticle is relatively impermeable to water and functions primarily to minimise water loss from the plant. The epidermis is also studded with these tiny pores called stomata. And stomata regulate water loss from the plant and enables gases to diffuse into the plant, into the leaves. And of course, the most important gas which the plant needs to take up is carbon dioxide for photosynthesis. All right, let's look at the structure of a stomata, or I should say stoma, one stoma, plural is stomata. All right, so what we're looking at here are epidermal cells of the leaf, and epidermal cells generally lack chloroplasts. They don't normally photosynthesize. But here we see one stoma, which we can still refer to as a stomata, and it consists of a central pore that can open or close, and the opening and closing of that pore is facilitated by two cells called guard cells. That's one there, one guard cell there, and here is the second guard cell. And it's the action of these guard cells, and we'll talk a lot more about this mechanism in living systems too. It's the action of these two cells that enable that pore to open and close. So obviously when it's closed, then that prevents loss of water vapour from the plant, but it needs to be open so carbon dioxide from the air can diffuse into the air spaces within the leaf, and from there into the cells that require carbon dioxide to carry out photosynthesis. So they're our stomata, and we'll see those also on this slide. So here's the leaf anatomy of a young leaf of Eucalyptus globulus, and we can very clearly see now this upper epidermis layer, and those epidermal cells will secrete that waxy layer called the cuticle. And here we have the lower epidermis, and it's in this lower epidermis layer that we can actually see one stomata there and one there. So primarily the stomata are located on the lower surface, but you do get them also on in the upper epidermis as well. All right, let's look at the important photosynthetic cells within the leaf. So the first type of mesophyll photosynthetic cell is called the palisade mesophyll, and they're normally found towards the upper side of the leaf. They're very long pillar-like cells stacked closely together, and most of the chloroplasts involved in photosynthesis are found within these cells. So they are packed full of chloroplasts, palisade mesophyll cells. Then in this lower part of the leaf, we find these very irregular shaped cells sort of scattered around, and these are called spongy mesophyll cells. And they also contain some chloroplasts and also function in photosynthesis. But also important to note are all these air spaces, particularly between the spongy mesophyll cells. This is where carbon dioxide will diffuse into these air spaces, and it diffuses along a concentration gradient, a carbon dioxide concentration gradient. From there, carbon dioxide follows another concentration gradient from these air spaces into the cytosol of these photosynthetic mesophyll cells to enable photosynthesis to take place. Right, to the right, we can see a mature leaf. Once again, same sort of structures, but now we can see one of the vascular bundles within the leaf that contains xylem and phloem. And we can see this as a leaf vein. Here we also see a specialised cell within the epidermis, and of course this is an oil gland cell. Being eucalyptus, that makes a lot of sense. All right, let's recap and add a little bit more to that. So leaves, special functions of epidermal cells in leaves may include the formation of leaf hairs, which function to minimise evaporative water loss from the leaf. Secretory gland cells, so we saw that oil gland in the leaf on the previous slide. Some cells will contain crystals. So for example, there's some species of grasses, if you run your hand along the blade of the grass, it'll feel very rough, and this is because of the crystals contained in those epidermal cells. We've looked at mesophyll cells. Mesophyll cells represent the ground tissue of leaves, and they comprise chloroplast-containing parenchyma cells that function in photosynthesis. Those mesophyll cells are often differentiated into the palisade mesophyll towards the upper surface of the leaf, and spongy mesophyll in the lower part of the leaf. Veins or vascular bundles of flowering plants form an interconnected branching system with xylem on the upper side and phloem on the lower side of the bundle. Mesophyll cells are often referred to as paraveinal mesophyll cells because they transfer sugars or photosynthates from mesophyll cell to mesophyll cell until those photosynthates can be loaded into the phloem vascular tissue within the vascular bundle. Our last slide for this lecture. Let's look at some modifications of leaf structure. You can imagine that there are aspects of leaf morphology that are directly influenced by the environment. So, for example, the difference in the structure of a leaf that's found in the shade, that grows in the shade, versus a leaf that grows in full sun. Shaded leaves, or leaves of plants that just grow in the shade, are generally larger, but have less palisade mesophyll than leaves that are found in full sun. Plants growing in particular habitats also exhibit leaf adaptations. So, for example, many of the Australian plants growing in arid and semi-arid regions often have very small scale-like leaves, obviously to reduce water loss. They have very thick cuticles and also the presence of lots of leaf hairs. And one final feature of leaves, and these are modified stomata, pore-like structures called hydothodes. Many plant leaves have them. They would be found on the tips, on each tip of the leaf. This is strawberry. And these little pores, they don't open and close like stomata do. They're just constantly open, and they enable what's called extrusion or guttation. That is water that's coming up through the xylem under certain environmental conditions, basically pushes out through these little pores. And it can be seen as tiny little droplets on the edges of the leaf, often seen first thing in the morning. And this generally occurs under conditions of high atmospheric humidity. And that's it for this lecture. Who needs a break?
Yeah.
Yes?
Yes, please.
All right. Would you like 5 minutes or longer?
5 is fine.
Okay. All right. I'll see you in 5 minutes.
Okay. Well,
Hey, Brad, stop teasing us and give us a look at that fish tank. You there, Brad? Better unmute yourself and tell me what you got in there.
Can you hear me?
Yeah, I can.
Yeah. So I got 6 goldfish.
Oh, yeah?
You can't get about as good of an angle I can get, unfortunately, because the cable's not long enough.
They look very pale. Oh, that's the end of the tank.
Yeah.
Yeah, cool. And what do you got above?
So yeah. So got a bit of a herb garden up the top there. Basil, coriander, chive, oregano, thyme, sage, and trying to think what the last one was.
Is it aquaponics or is they just growing there?
It's aquaponics.
Cool. Very good. Yeah, no, you're teasing me the whole time showing me the end of a fish tank. That's just mean.
Yeah, unfortunately where it is, it's on the end of my computer desk.
Yeah.
So cable's not long enough to really bring it to the front, unfortunately.
And how are you finding your aquaponic system? Is it working well or not?
I've had a bit of an iron deficiency, so I topped that up today. That's why the water's a bit off colour. Between that and a little bit of sea salt, the water's a bit brown at the moment. But
Very brown, yep.
But it's been clearing up slowly. But it seems to be going well. Like, nitrate levels are low-ish, but they're there. No ammonia or anything, which is good.
Mm-hmm, yeah. Nitrate's okay. It's the nitrite that'll kill your fish pretty quick, or ammonia that'll kill your fish really quick.
Yeah, well, there's 0 of both of those, thankfully.
That's good. I wish I could say the same. All right. Thanks for giving us a look-see.
Yeah, no worries. I wish I could show you from the front because it looks much more impressive from the front, but yeah, the cable's just not long enough.
No, fair enough. Fair enough. All right, mate. Catch up with you one day.
Yep, will do.
Okay, is everyone ready to go?
Yep.
Yes. Okay, Brad's back. You were having a good chat there, Brads.
Yeah.
I heard something about fish tanks going on in the background.
Yeah, and he was asking about the little fish tank I've got on my desk at the moment.
Oh, right.
I didn't realise my webcam was on and it's focused on the fish tank.
Oh, no, it's good. It's good. Very distracting for me. Oh, yeah, I can see it. What have you got in there, Brad?
So I've just got a couple goldfish, and then I've got just a few herbs on top.
Oh, right, okay.
So it's just a small aquaponics setup.
Oh, very nice.
Flows up to the top and back down.
So have you got something growing in that aquaponic setup?
So I've got a bunch of herbs. I've got sage, coriander, chives, thyme, basil, and oregano.
Ah, fantastic. And where's the light source?
Usually just comes through the window in the back there.
Ah, right, okay.
I do want to get some proper lights for it to help them out a bit, but
Yeah.
I'm just relying on the sun.
Okay, and I suppose you can't have that outside, can you? Not with goldfish, no.
Yeah, no, not really.
They wouldn't like it outside. Oh, that's terrific. Aquaponic setup.
Yeah, it's just a small little one at the moment.
Yeah, no, that's great. That's terrific. The way of the future.
Yeah, pretty much.
Yeah, I think so.
It's just a shame that the water's not looking a bit better at the moment. I had to add some iron into the system, so I was kind of discoloured the water.
Oh, right. That's why it's looking a bit brown there.
Yeah. I might have added a little bit too much.
Maybe, yeah. But the fish is still happy, so that's
Yeah.
good.
They seem happy. They've just had a bit of a feed earlier, and they seem to be good with that. So
Fantastic. No, that's great. Thank you for sharing that with us, Brad.
no worries.
All right, let's move on with lecture 15. And as I said earlier, we're not covering the whole lecture. We'll finish this next week. So we're going to be talking a lot more about DNA. So let's start with one of our major biological macromolecules, the nucleic acids. And you remember the two major types of nucleic acids are DNA and RNA. So we're going to revise what we've learnt so far. We know that nucleic acids are found in all living cells, and that the molecules in which information on cell structure and function is stored and transferred from generation to generation is DNA. DNA is mostly found within the nucleus of the cell, or most of the DNA is found in the nucleus of the cell. It's never found in the cytosol. There are very tiny bits of DNA found in certain organelles, such as mitochondria, in all cells, and in plant cells, organelles, the organelles called chloroplasts, also contain tiny bits of DNA. Building block of DNA, as you know, is the nucleotide, which has three parts: nitrogenous base, a monosaccharide, which is either ribose or deoxyribose, and a phosphate group. So let's recap the structure of a nucleotide. Once again, base, sugar, and phosphate. Let's look at that again. So this particular base shown here is just one type, one of 5 types. This one's called adenine. That is attached to the monosaccharide, the sugar, which is shown here. This particular sugar is ribose. So a nucleotide that contains a ribose sugar would be found in RNA or DNA. Who remembers this?
RNA.
RNA, yes. Yep, so the sugar in nucleotides of RNA is ribose. If it was a sugar found in a nucleotide of DNA, it would be 2-deoxyribose. And of course, we have the third part of the nucleotide, the phosphate group. Now, let's look at the numbers here in the actual, in the sugar. 1', 2', 3', 4', 5'. These numbers refer to the location of the carbons. So they're not shown here, but here there is a carbon, and we call that the 1' carbon. There's also a carbon here that's not shown. That's the 2' carbon, 3' carbon, 4', and the 5' carbon is indicated here. So we can see that the 5' carbon is where the phosphate group is attached. So we call this the 5' end, the 5' end of the nucleotide, or we refer to this as the 5' phosphate, because it's attached to the 5' carbon of the sugar. Here we have the 3' carbon, and attached to that 3' carbon is a hydroxyl group, this OH here. So this is referred to as the 3' end of the nucleotide, and this becomes important later on. 3' hydroxyl and the 5' phosphate of a nucleotide. At this 2' carbon position, this is where ribose differs from 2-deoxyribose. So in ribose, which we see here, the 2' carbon is attached to a hydroxyl group. But if this was deoxyribose, then instead of a hydroxyl here, we would simply have a hydrogen. All right, so that explains why RNA is called RNA, because it stands for ribonucleic acid, in other words, ribose. And DNA is derived from deoxyribonucleic acid. So that's referring to the deoxyribose sugar found in nucleotides of DNA. right, the nitrogenous bases are of two types, pyrimidines and purines. So the pyrimidines includes 3 bases and therefore 3 nucleotides that contain these bases. They are called uracil, cytosine, and thymine, abbreviated to U, C, and T. And they are called pyrimidines because they consist, the base consists of only a single ring of carbon and nitrogen. We have 2 purine bases or 2 purine nucleotides called adenine A and guanine G. And these consist of 2 rings of carbon and nitrogen. Okay, you really don't need to remember pyrimidines and purines, but you certainly should be familiar with the 5 nucleotides: uracil, cytosine, thymine, adenine, guanine. All right, let's revise. In DNA, the monosaccharide is 2-deoxyribose, which means that the hydroxyl at the 2' carbon position is replaced simply with a hydrogen. The bases, and therefore the nucleotides found always in DNA, are adenine, thymine, guanine, and cytosine. RNA has a ribose monosaccharide, and the bases or nucleotides are always adenine, uracil instead of thymine, guanine, and cytosine. So let's revise our base pairing rules. Who remembers what they are? Base pairing rules in DNA. A always pairs with, does anyone remember? A always pairs with T, and C always pairs with G. Okay, and we'll see that in just a minute. Oh, here we go, on the next slide. So you've seen this slide before. To the left here, we have our DNA double helix, and that's shown again here in this image, but now we can actually see how the nucleotides are paired up between one strand of nucleotides and the opposite strand. So the G always pairs with C on the opposite strand. A thymine on one strand of DNA always pairs with A on the other strand, and so on. And you can see that the bases of each nucleotide are found towards the centre of this double-stranded DNA helix. These ribbons here, the pink ribbon represents the sugar phosphate backbone in one chain of nucleotides, and the purple strand represents the sugar phosphate backbone in the opposite strand of DNA. All right, so let's look at that in more detail. We'll take this boxed region here of 4 nucleotides and enlarge that so that we can see all the detail. All right, so we'll start by looking at these are 2 nucleotides that are found on one strand or one chain of DNA. And remember, this is only a minute portion of all the nucleotides you would find in one chain of DNA, which we call a strand of DNA. There would be something like hundreds of thousands or even millions, depending on the organism, of nucleotides in each strand. Okay, so we've got a cytosine shown here, and in the same strand of DNA attached to it, we have a thymine. On the opposite strand of DNA, we have a guanine and an adenine. The 2 strands of DNA are joined together, as you know, as a result of hydrogen bonding. There's one hydrogen bond there, here's another one, and here's another one. There are always 3 hydrogen bonds that form between cytosine and guanine. And this determines those base pairing rules. This is why cytosine and guanine are always paired up together on those opposite strands. If we look at the hydrogen bonds that form between adenine and thymine, there's one there and one there. So always 2 hydrogen bonds that form between those two.
Silvana,
Yes.
I thought hydrogen could only have 1 bond. Did you get it or not? I thought.
Yes. So you're talking about 1 covalent bond.
Yes.
Yes, that's correct. Hydrogen can only form 1 covalent bond, but these are not covalent bonds.
I remember you saying that now.
Hydrogen bonds are not covalent bonds. Yeah. All right. They're just interactions. Or who can tell me?
Are they intermolecular forces? Or what is that?
Sorry, say that again.
Intermolecular
Forces, yes. So these are just attractions, yep, intermolecular attractions between hydrogen and a more electronegative atom on another molecule. So they're not covalent bonds. So we've got, remember we talked about delta negative and delta positive, these partial charges. And that's the nature of the hydrogen bond. Okay, good question. All right, so that's clear that. All right, now remember that we said that the carbons on the sugar are numbered from 1' through to 5'. Here's the 5' carbon, which attaches to the phosphate. So we refer to this as the 5' phosphate. That means that this end of the string of nucleotides, the chain of nucleotides, is referred to as the 5' end. And if we look at the other end of that chain of nucleotides, we can see the 3' hydroxyl. That is the hydroxyl group that's attached to the 3' carbon of the sugar. So we call this the 3' end of this chain of nucleotides. Let's look at the same thing on the other strand of DNA, and these nucleotides are upside down relative to the opposite strand, so the ends are also opposite. 3' end up here, 5' phosphate is down this end. So the 2 strands of DNA are said to be anti-parallel as a result of that. Okay. DNA is organised into structures called chromosomes. Each chromosome is a single DNA molecule. That is 2 strands of DNA joined together by hydrogen bonds is referred to as a single molecule of DNA. In eukaryotic cells, chromosomes are always located in the nucleus, and they are characteristically linear. This is in contrast to chromosomes in prokaryotic cells, such as bacteria, where the chromosomes tend usually to be circular rather than linear. Each species of organism has a unique chromosome complement, that is, a unique number of chromosomes in the nucleus of each cell, shape of chromosomes and size of chromosomes. Just a couple of terms here. The centromere, a region called the centromere, is located in the middle of the chromosome, and this is essential for cell division. The 2 ends of each chromosome are referred to as telomeres. Okay, if you were to extract all the chromosomes from the nucleus of one cell and arrange them as they've been arranged here into pairs, you would have what's called a karyotype. So these are actual chromosomes that have been extracted and arranged as such, and they've been extracted from a cell of a human, male human. And we can see that there are 22 pairs of chromosomes, plus an additional 2 chromosomes, which are called the sex chromosomes. So, of course, in males, the presence of an X and a Y chromosome determines maleness, determines gender. If those chromosomes were extracted from a female, then the sex chromosomes would be 2X sex chromosomes rather than the X and the Y. All right, so you can see that we have pairs of chromosomes, 2 copies of chromosome number 1, 2 copies of chromosome number 2, and so on and so on. All right, we have the same thing here, karyotype from a female, but this female has the condition Down syndrome. And what causes Down syndrome is the presence of 1 additional copy of chromosome number 21. So, of course, being able to replicate chromosomes precisely every time a cell divides to produce two new cells is critical, super critical. 1 mistake and this is what can happen, an additional chromosome. In this case, it's not a mistake that that is fatal to the developing embryo, but of course Down syndrome comes with a whole host of problems, heart problems and so on, intellectual disability, etc. All right, so that's a karyotype of human chromosomes. And of course, even though every nucleus of every cell contains exactly the same chromosomes, the same complement of chromosomes as what we're seeing here, what makes different cells function differently is that the genes contained within these chromosomes are expressed differently in different cell types and different tissue types. Okay, eukaryotic chromosomes are made up primarily of proteins and, of course, DNA. The proteins are called histones. They assemble in groups of 8 and form a core upon which the DNA is wrapped around, and together this is called a nucleosome core particle, or simply a nucleosome. Not particularly important for us, but we'll have a look at that on the next slide. So here we have a nucleosome particle, various types of histone proteins in the middle there. In blue, this is the DNA, which is wrapped a couple of times around each of those groups of histones. And there's 1 histone also down here. So each chromosome contains the DNA and proteins. All right, let's look at our double-stranded DNA here, just on its own. And if we follow that down here, we can see that it's going to start wrapping around these histone proteins. Here it is, wrapped around. And now we condense and coil that DNA and protein even further until it becomes part of a chromosome, forms a chromosome. What we're seeing here are 2 chromosomes joined together at the centromere. And this is something that occurs during cell division. All right, and that's about as far as we need to go tonight. So who has questions for me?
No question, just a statement that when I woke up this morning, the news was that they found a new DNA, and I thought that would be typical. And the day I'm about to do DNA, they've changed the rules.
They have found new DNA.
Yeah, one that they fluoresced or something, and it turns on and off. It's on the news this morning.
Right, okay. I need to find that. That sounds fascinating. Thank you for that, Liz. All right, so here are the tutorial questions based on tonight's lecture, Structure of Plants. They are not assessed, but of course, as always, please work through them very carefully, and you can check your answers via the non-assessed online quiz. Okay, and everyone should have received their mid-semester test results by now. This is just a spread of the marks. Yeah. If anyone wants to know their exact mark for the mid-semester test, just send me an email and I can tell you what your exact percentage was for the test.
I think it appears on Moodle, Silvana, you can see.
Yeah, pretty sure it does.
It does?
Yeah.
Yep.
Your exact mark or just the grade?
A percentage?
Oh, you do get the percentage?
Yeah, you get both.
Oh, good. Oh, terrific. Okay. Oh, there you go. You don't need to send me an email then.
One less email.
All right, that's terrific. Any questions about your prac reports?
No.
No? So hopefully everyone's made a start, at least on one report.
Yep.
Please remember with referencing, if you're not that comfortable with referencing, go to the Ernie Easy Referencing site on the library website, or you can just type into Google search Ernie Melbourne Polytechnic. It should take you straight to Ernie Easy Referencing. Okay, really easy way to learn how to reference correctly. And, you know, I'd really like to sort of stress how important it is for first year students particularly to get into the habit of referencing according to Harvard style. Because come third year, there's still, I think, too many students that don't do it properly. And it's really important that you've developed good habits so you're not constantly losing marks on assessments and reports just because you're not referencing correctly. Okay, so Ernie Easy Referencing, ERNI. And if you have any questions about that, please let me know.
Silvana, when referencing the prac manual itself, because I've seen different ways that it gets done, because it's a bit difficult just with its format, is it most important that it's just there rather than the exact way that we reference it?
Yes, that's right. Correct. It's important that you just include the reference, but remember also that you shouldn't just reference the prac manual.
No.
Important that you use other references, at least 2 other references in your report. And also remember one thing that some students seem to not follow was the word count. So the word count for the prac reports is 500 words, but that word count only applies to the introduction and the discussion conclusion section. So I would expect to see at least, roughly 200 words in the intro and 250 words in the intro aim section and 250 words in the discussion conclusion section. I don't mind if you go over that word count in those 2 sections, but you certainly shouldn't be writing only 50 words in each section. That's just far too brief, and you would lose marks as a result. So it doesn't matter how long your methods or results section are. It does not matter. And the references, of course, are not counted in that word count. And having said that, of course, always check when it comes to word counts with your lecturers. Some lecturers are very, very particular about word counts, so they don't want you to go more than, 10% over or under. Otherwise, you'll start having marks deducted. For my reports, I don't mind you going over, but certainly not under the word count or too far under the word count. And remember, if you'd like to email a draft of your first report to me, I can give you some early feedback before you finish writing your second report, and of course before you submit those reports. So feel free to send that a draft through of your first report, but please make sure that you send it to me at least 1 week before it's due. Otherwise, I can't guarantee that I'll have a chance to look at that for you. Any other questions?
No, but
No? All happy?
Found a link to the article that Liz was talking, or an article that.
Fantastic. Thank you very much, Darren. A new form of DNA in our cells. Fascinating. I'll be looking at that straight away.
Enjoy with your dinner.
Yes, thank you, with my dinner. Absolutely. All right. Thank you, everyone. Have a great week.
Good night.
Yep.
Thank
All
you.
right.
Thanks,
and I'll see you
Ivana.
next week.
Thanks,
Thank
everyone.
you.
See
Okay.
ya.
Bye-bye.